Let me introduce our distinguished speaker, Dr. Khalid Naeem Khwaja. Dr. Khalid Naeem is a One Health expert who completed DVM in 1976 and MSc in microbiology in 1980 from the University of Veterinary and Animal Sciences, Lahore, Pakistan. He did his PhD in virology from the University of Minnesota, USA in 1990. Dr. Naeem has served for 35 years at different research, teaching, and administrative positions at the National Agriculture Research Center, Islamabad, Pakistan. Studies and control of emerging pathogens, especially zoonotic in nature, have been his major area of work, where he initially coordinated to set up and run a national avian influenza control program between 2002 and 2010. This has led to the successful control of H5N1 in Pakistan. Later, in collaboration with the Massey University, New Zealand, and NIH, his research team set up the first One Health Pakistan network in 2012 for coordinating the surveillance of major zoonoses, such as CCHF, brucellosis, and influenza. Dr. Naeem is an approved WHO-certified biosecurity auditor, possessing experience of evaluating some specific pathogen handling facilities around the country. He has served on several national policy formulation committees, including AMR containment, zoonotic disease management, and national laboratory policy development groups. He has supervised research of 45 MPhil and 10 PhD students and published more than 90 research articles in peer-reviewed journals. He is currently engaged with private sector in diagnostics and animal vaccine production. Dr. Naeem, sir, is basically an institution in himself and an inspiration for many people like me. Sir, absolute honor to have you.
Thank you very much. Appreciate your introduction, and thank you for having me in this kind of discussion. And I think let's proceed. You have the presentation. Maybe you can start with it, and I can start discussing it. I think as the topic, today's topic basically depicts that we are going to discuss about the new issue of H5N1 outbreaks in dairy cattle. It's very recent subject, and there's very little information available in literature about the current situation. But based on our experience of dealing with avian influenza since 1995, I think I can have some sharing of experience of those avian influenza outbreaks as a little bit background, how it can affect the animal side, like the dairy cattle, and how important this is for the public health issues, like because of a zoonotic disease, how can this somehow affect or get involved the public health issues? And that's what we'll be discussing in our question answer session as well. So next slide, please. I think most of the participants would know what zoonoses are, but just for the sake of revising it, we are talking here about the diseases of transmissible nature, which has been defined by WHO, that the disease is transmittable from vertebrates animals to human and vice versa. So that's what's defined as zoonosis. And there's more than 200 described pathogens which have been incriminated in such type of situations in which these infections transmit between human health, and animal health sector. Just to review what has happened in this century, 21st century, I think many of the participants would be pretty young being active in this century, 21st century, where we have seen this the occurrence of this West Nile virus in the early days, very late 1999 and 2000, and from there onward we have seen number of outbreaks listed over here, number of diseases which were, a couple of them were old kind of bugs like anthrax, and a few of them were very new to us, like SARS, and the influenza is it's not very new, but if we talk about the avian influenza transmitting to human, it has happened in the past century as well. But as our focus of today's discussion is H5N1, so I would just like to mention that the number of outbreaks have happened in this century, and the most important was in 2003-04, when the H5N1 was first time basically spreading at a high rate to almost most of the countries in the world, and there was transmission to human was also noted in that period. And then later on, the COVID, we have faced COVID, and maybe people will be surprised to seeing it zoonosis, But as it is, very well known that bats are the host of this pathogen, so we basically place it in this category. And the last one which we are discussing today is H5N1, which is a very recent event, and we'll be discussing it further. And apart from this, number of other small events like the Lassa fever, the Nipah, the Hendra, and other pathogens causing these kind of infection have also surfaced in the past couple of years, but we do not have much information because they were isolated incidents, and they were not kind of a panzootic kind of thing, or a pandemic kind of situation. Just generally to give you an idea about the viral emergence, as we know that like other pathogens, viruses have also got the capability to evolve, genetically modify, their genetic material reassort. There's some point mutation at some time, recombination at other time, and a number of things happen in the field whereby we come up with new pathogens. Sometimes the previous organism, they emerge as a new strain, a new genotype or clade or subtype, and sometimes because of the combination of various strains, we evolve a different kind of virus which was not noticed before. So we see in the present time that because of the use of the heterologous vaccines, both in animal health and human health, we have never been able to control completely an infection, a virus, which is inherited abilities of mutation. And the influenza virus is one of the good examples. Because of the continuous mutation evolving, there's new strains which evolve, and that's how we see the introduction of new organisms or new mutant of the same virus. Similarly, by using excessive live vaccination, that is one of the factors by which the environment gets contaminated, and thereby it helps in generating new mutants from the same virus circulating in different forms in the environment. May it be the live virus from the vaccines, may it be the live virus from the same group but a field virus. So this new combination also helps generating new mutants. Other issues in animal sector are primarily the biosecurity issues, whereby the weak and pure biosecurity basically helps in introduction of the virus escaping either the vaccine virus into the environment. Environment, when I say, it means the wild animals like birds and rats and mice, and that's how the virus will escape. Similarly, they will also be involved in introducing the pathogen from outside. Similarly, disposing of those dead birds, dead carcasses, that's another source of infection. So that also helps in generating new organisms. Another flaw is the lack of diagnosis, and I always say late diagnosis. They're two different things. One is we do not do any diagnostics and have kind of a therapeutic approach, and one is that I do the diagnostics, identify the pathogen, and then carry out proper surveillance for a long period of time, and then evolve a control strategy. So unless that strategy exists, we keep on seeing new mutants despite having vaccination or despite having new introduction of vaccination in new areas. And the last thing is in this category would be the regulatory mechanism which we have that also somehow affects negatively to the generation of these mutants. Now let's talk about the avian influenza viruses, which is the topic of today. Generally, there are two types of viruses based on their pathogenicity. We classify them into low pathogenic avian influenza and high pathogenic avian influenza. And their type, one is the HA column. To type it, it is the HA protein, which has 1 to 16 different types of molecule. And similarly, the other protein is NA, neuraminidase protein, that has 9 types. So with these combinations, almost more than 300 combinations are possible of avian influenza viruses of different types. But the two major components, the low path and high path, are defined. In case of the low path viruses, they may belong to all these 16 subtypes of H, and they may not cause any major disease, but they will be producing mild infections in poultry. And in case of high path influenza viruses, we primarily consider H5 type and H7 type as a high path. because they are present basically in both forms, low path and high path. But the low path switches to high path, and primarily there are two major components so far known out of all these 16 subtypes of H. So if you give this combination name, I've just given one example, H5N1. So it will be taken H5 from the hemoglobin antigen, and from the neuraminidase antigen. So thereby you have a combination of the virus, and the current virus is H5N1. And when you genetically do sequencing and classify these viruses, currently by the genetic lineage approach, we have the grouping of the is 2.1.3 and so on. It continues with the mutations, and the current clade, or can say subtype that we have, it has a number 2.3.4.4b, which is basically causing high mortality in wild birds, and that's the virus we're going to talk today. And this is the example of basically different, you see, name of different viruses, if you see at the end, which we have found in the past many years affecting the animal population with the H5 combination. You can see H5N1 here, H5N6 here, and H5N2 here. So these are different outbreaks, different isolations, and just to give you a feeling that the variety of H5 basically has been present in the environment in the world, all over the world. Now, the currently focusing on avian influenza in animals, currently I borrowed this slide from the World Organization of Animal Health site, and this is to depict, you can see the spread of this H5 influenza viruses all over the world. So in most of the countries, these viruses, the H5 viruses have been prevalent since basically 2021. So from 2021 till to date, a variety of outbreaks have occurred because of H5, primarily H5N1. But some other H5N8 and some other subtypes have also been reported in this period. This is a source, the Australian source, which just have given some data, which indicates that during these years from 2020 to date, we have around 85 cases of human infection with this H5 type. And it may be H5N1 and maybe H5N6. If you see it, which is different, subtypes have been causing infections in animal and later on infecting the human. That has been the trend. Now let's focus more on the H5N1 outbreak in dairy cattle, current issue. Historically, this outbreak was reported way back in January in 2024, and the first time it was realized that there's a problem with the dairy farms, which has the major issue of sudden decrease in milk production and some anorexia, like lack of eating of the food of animal, and this kind of uncomfortable position of the animals. Otherwise, there was no specific clinical condition which can be correlated with any specific disease at that time. But later on, when more investigations were carried out, it was found out that this is the H5N1, and further studies indicated that this is the same H5N1, which was already prevailing in states and other countries and Europe and other South American countries in wild birds and also in wildlife. Like many other animals, many other types of mammals have also been affected by H5N1. So later on, just in last month, April, WHO identified and reported first human infection because of the same H5N1 strain of virus, which spread out from that farms which was infected with H5N1, or whether dairy was infected with H5N1. So that was the first report of human infection from that part. Later on, more studies were carried out to see the spread of this infection, because as this first time you come to know if there's infection, that there's always a way to go back and study the previous old serum samples in the storage bank. They go and see the food products produced from animals. They want to see the other farms which are present, and they don't report anything, or they have not reported anything, to have a clear picture. Still, it's very early, and there are a lot of studies are ongoing. Every day you see a new report, so it's very difficult to comprehend and conclude anything. But we just try to see the current situation and try to assess and suggest the way forward. Based on this situation of these dairy, more and more dairy farms have been found affected, and currently the risk assessment evaluation by WHO has revealed that the current virus, the current sequence of the virus, the other characteristics identified and studied, they reflect that this virus has a low kind of risk posed for human health in the current situation. That's the conclusion from WHO. As I'm saying, this is for today. How it evolves or how it moves forward is still yet to be seen. And the risk was basically dependent on a couple of things. It is for those people, those workers who have been, exposed to the infected birds or animals with a currently circulating H5N1. The risk of infection is still considered to be low or moderate in sense. However, as I said, as the things evolve, new things happen, virus continue to mutate, evolve. This risk assessment has to be carried out repeatedly in coming times as well. Some other studies about the milk and other products have also been reported, and it indicates that milk from these animals was carrying this virus. But as the milk is pasteurized in states, so at most of the places, the pasteurized milk has been considered to be less risky, and generally the thought is that the food products produced from these animals will be safe. However, the unpasteurized milk used by animals like cats and some other situation, that was found still contaminated, and that infected those cats. That's also reported, and they produced some serious condition in those animals. And further investigation is going on. What's the effect of those contaminated milk to the other mammals which are going to use that milk. As far as the infection spread is concerned, it is still not clear how the infection has taken place. There are a number of hypotheses and theories about it. We still don't know the exact transmission pathway of this virus. We still do not know primarily if it is spreading from cattle to cattle as a virus and through which media, or maybe it's at least we know it's not airborne. So if it's not airborne, then maybe there are some other things to look into. Some people are suggesting that maybe contaminated feed may be responsible through the infection by the wild birds, or their fecal material contaminating the feed, and then feed going to the cattle, and that's how they think this is spreading. Some mechanical means are also being investigated. The previous history is that a number of times, like maybe 3 or 4 times in record, the infection of the influenza virus to the cattle has been reported, but that has not been, there's no report of having a very serious impact on the health of the animal. So that's kind of still at very early stage how we can define the current outbreak. So keeping this situation in view, the USDA, they have advised 2 things: primarily mandatory testing for interstate movement of animals, and also for testing the influenza in livestock and reporting that to the central place, USDA. Historically, WHO is taking care of the surveillance of influenza viruses generally, and those influenza viruses which are basically affecting the human, and the report says that there are around 800 detections of human infection since 2003. As I mentioned earlier, that the outbreak of H5N1 from 2003-4 onward, it stayed here for the next, I think, 7 or 8 years. In those days, the very high report of H5N1 infection, animal and human as well. So this is what all data indicates, that from this much bigger period, 800 detections were reported. The clade, which we are talking right now, the 2.3.4.4b, it is basically continuously evolving. A lot of sequencing work is being reported, and it reflects that still the range of this virus is the wild animals, wild mammals, and wild birds as well. Let's focus more on the H5N1. So during, as I said, the recent outbreak started in 2021 in birds, and from there it spread to other mammals. During this period, 28 human infections have been reported. Among these cases, the good thing is there's no report of human-to-human infection transmission. So the most important thing in influenza, in this kind of influenza viruses, is to determine if it is capable to transmit from one host to the other host, like from one mammal to the other mammal, and especially from either animal to human and then from human to human. So unless we establish a chain from human to human transmission, there's the risk of converting it into a pandemic is very, very low. But that's what the theory says. However, our effort of investigation and sampling and surveillance continues to understand if there is any mutation which may lead to this particular phenomena, and we know scientifically what mutation is specific for a virus to enable it to transmit from human to human. Second here to mention is about the swine influenza viruses. These are one of the viruses which stay not only stay in swine, but they're also transmittable to human. And the swine serves as a mixing vessel in which the infection comes from the birds and comes from the human, and the both viruses mix together there and may evolve a new virus. That's one of the good example of a mixing vessel, animal as a host, which act as a mixing vessel to transmit, not only transmit the same virus, but to help in evolving a mutating virus, which may be more virulent to the next species, maybe like human in case of human. So that's what we are always afraid. As swine is still working, doing that job. So based on this available data, the WHO has evaluated, and the joint effort of the WHO, FAO, the World Organization of Animal Health, they have now jointly listed a few, factors, a few assessment criteria. So at present time, based on this information, as I said earlier, public health risk is relatively low. It's low for those with exposure to infected birds or animals, and there's no reported human-to-human transmission. So, generally speaking, the risk overall is considered less. However, as I said earlier, there's something repetition over here as well, that this risk assessment need to be continued in every environment, in every country, in every situation, so that anything happens new is to be noticed, and then the rate of risk will eventually be modified accordingly. There's some other concerns, scientific concerns about this virus, which I just wanted to list down. It's not to create fear, but it's to basically share that what people are thinking, what scientists are thinking, which way this virus may go. As I said earlier, the current detection of the dairy cattle issue, the other mammals affected and reported earlier during the past few years have been these minks, the sea lions, and variety of human cases, as mentioned earlier. Still, the new virus is bird adapted, and it is still not transmittable from even mammals to mammals. So that part is keeping us a little bit, you can say, quiet, and it's basically helping us to work more to understand this viral structure. So before it adopts this habit of transmission from mammal to mammal, and then, God forbid, from man to man, that will be a kind of terrific situation. So, as we know that so far there's no mammal to mammal transmission, no man-to-man infection. So the concern are that, the concerns were about the pasteurized milk that as a source of transmission have also been subsided because of some early studies in US. So that part is somehow taken care of. However, there is no sign of any decrease in outbreaks of cow in states. And some other scientists are increasingly concerned that if the cattle becomes a permanent reservoir from this adapted virus, then there's more chance of its mutation. As I said earlier, that once the virus circulates in a host and keep on propagating, it adjusts and modify, mutate in a way that it becomes capable to jump into human, like I said about the swine case. So that's a fear. And that fear is for the scientific community and obviously fear for the public health at large scale. Another data indicates that virus is basically shifting between birds to cow right now. And the cow may work as a mixing vessel, like I gave you example about the pigs, So the fear is that if the virus establishes in cow and the cow become a normal carrier of this virus, so it will be a major source of infection transmission to the human at some stage or some to the species. Even if the virus is not a killer for cow, it may become a permanent carrier like birds, wild birds, like pigs. I'll give you two examples. So some other data for the 200 viral genomes taken has found that the virus has jumped from wild bird to cattle in somewhere in late 2023. So you can well imagine the power of sequencing and the power of analyzing the genetic code, genetic makeup of those viruses, how important it is to study all those viruses which have been circulating. And they have identified for when first time this virus entered in cow. It's almost 1 year later. Now we come to know that it has infected cow. So important thing is that once the virus transmission becomes understandable, right now we don't know how it has been transmitted. It is not respirated out. So once we know it, it then will be possible to develop an effective control strategy, which may also include vaccination, obviously, in case of animals, I'm saying over here. So after this kind of background information, a few slides for the strategy, how to develop some control measures. As I mentioned you earlier, we had experience of handling H5N1 in Pakistan also. So I'll give you small, few slides for that. So this is a general cycle of general disease management. You have to detect it, respond it, and once you respond, you have to recover. You have to plan for next prevention, and then again the cycle goes on. So this cycle of disease management is to be applicable for HPAI influenza as well. So the standard response strategy, which we basically used in Pakistan during 2005 to 8 outbreaks, has recently been revitalized. Last year we had a meeting, and the previous strategy is called National Contingency Plan for the containment of HPAI. It has been revised now. So these are some of the salient features which have been listed in that policy. As I said, detailed program, very comprehensive. And interestingly, we have applied it way back in 2005 to 8 and successfully controlled H5N1 in Pakistan in poultry. And since 2008 to date, there's no report of H5N1 in Pakistan. So that's another detailed discussion, how it happened and what are the good things we have considered in this policy. But as this current issue is more inclined, more related to the livestock like cattle, so we'll have to modify this strategy. Different components of the strategy will need to be modified to accommodate the needs of the cattle management in this country. It's a kind of detailed policy. I don't want to spend more time on it, but in case someone has questions, we can discuss it. Policy also says that we need to have a good collaboration between animal health sector and human health sector. Zoonotic, generally, HPAI, this influenza hypath, specifically, are one of the good examples of One Health strategy. And what is this One Health strategy? It indicates basic collaboration between the animal health sector, human health sector, and environment. And this listed chart and this listed pathway is basically a strategy whereby we can work together and plan a format whereby we can encounter this organism or this type of organism more effectively. This is a successful model. The past, I'm talking about 2003 to 2008, during that high path period, H5N1. This linkage between human influenza work, animal influenza work in the presence of WHO, OIE, FAO, these international bodies, and our own local sector can have a very effective model to successfully handle this particular situation. Now, this is the last slide, just to give you way forward. That yes, there's a problem. Yes, we are trying to understand what the problem is. Yes, it's not here in Pakistan yet, but it's not in other places in the world as well. But every country is devising a strategy, planning, and this is the listed suggestions for strengthening our laboratory capabilities, for increasing the surveillance, enhancing the zero surveillance in all animals now, dairy animals as well. Another meeting just last week, I suggested to take up some preemptive approach of screening the current herds, cattle herds, for the presence of any seroconversion. As we found in the USA case, that detection was done 1 year later before it entered in the population. So we can do is we can start it right now at this stage. And once we detect it, if there's a virus or maybe just serology, but once we have a seroconversion, we detect it, we type it, sequence it, and a lot of work to be done before we are able to understand how to encounter it. Because there are two strategies in the world for HPI. In the bird, it was in poultry, it was just check and slaughter it. In Pakistan, we adopted the strategy. Okay, you check the affected bird, the affected flock, slaughter it, but vaccinate the other in the surrounding. And that's how we have been able to successfully control it. Same will be done with livestock and other animals as well. And then we have to provide the specific material for culling of those animals, the carcass disposal, and some biosecurity measure involvement in case of the farm animal, because we know the coordination and collaboration of wild birds, the animals, the whole disease basically starts, and then human becomes the end up, you can say, eventually it ends up in that human infection, and the whole chain can be broken by working together. Another approach is the priority vaccination. Once we know that the virus is established in a species, maybe we can develop a vaccine for cattle as well. So that's not a problem. It's only the way we think, the way we plan. And last thing would be that we have to do some lobbying at higher level, national level, to revive the surveillance program, which was already taking place in the country, which was somewhat stopped because of disease was gone. As you say, some other disease came up, so that program goes behind the curtain. So it's high time to think about reviving the avian influenza surveillance strategy in the country. I think that's all. Thank you very much.
So can you explain a little bit more how does such a response is carried out in Pakistan? I know you mentioned that different stakeholders are involved. So how do the animal or the human sector or even the environmental sector, they coordinate in such situations in Pakistan?
That's a very important question and very important information to be shared. The past experiences that the Ministry of Health, Ministry of Agriculture, and Ministry of Environment, basically they made a combined body, and that committee was basically formulating these activities, and a number of plans are available for other countries, and we can always use our experience, past experience, and take the lessons from other people who have done work in this area and devise a strategy how to strengthen our disease reporting system, strengthen our disease surveillance system, sample collection, sample transportation, and then obviously lab investigation, rapid response teams. So all these things have been done previously already. So the protocols are available. The only thing is just to decide. We have to sit together and plan how to proceed and when to proceed. Otherwise, strategy is known. I mean, there are people around and there's documents around.
You mentioned about the surveillance. So can you especially explain what are the diagnostic facilities available at national level and at provincial level in Pakistan? Do we have the genomic sequencing capacity if we see a case of H5N1 in Pakistan in the future? So how quickly our testing or genomic facilities can do the sequencing?
Basically, as far as the animal health sector is concerned, there were basically two laboratories which have the sequencing setup and also the facilities for the PCR and virus isolation and typing. Otherwise, now, I was just in a meeting yesterday, at provincial level, they have set up, each province has got a central laboratory, which has the PCR and diagnostic stuff available, but no facilities for sequencing work. The federal laboratory in Islamabad, they have a sequencing setup, but still, as far as I know, it's not basically working right now because of the financial issues and maybe some other issues. So you're right, the diagnostic part is there, but sequencing and investigating further for typing and devising new diagnostic tools or new vaccine is lacking. Human health, I know there at your place, there's good facilities. You've done a lot of good work during this COVID. You have facilities. I think you're doing a good job. Maybe that can be also expanded at provincial level as well. Each province can have it so that we have to move fast. It virus moves very fast, honestly. So we have to go ahead of it before it spreads, establishes, mutate, and then we need a new vaccine. That will be a very difficult part.
Sir, thank you. Sir, you mentioned about the surveillance, and I will particularly request you to talk about the environmental surveillance because during the pandemic we have seen the importance of wastewater surveillance, either tracking the different variants of the SARS-CoV-2 or maybe giving a early sign. And so, what do you see? What is the importance of wastewater surveillance regarding H5N1?
Basically, it's very difficult part. Transboundary animal diseases, a very important component. Number of projects have been launched, but as our borders are porous, systems are not up to date. So, honestly speaking, I don't have a solution for that. But it needs a lot of work to establish rapid response units, those laboratories, rapid diagnostic labs, and at the point of entry in at the country, and also at the trading part as far as the animal products are concerned, or when we are importing material from other countries, the licensing. Like recently, I proposed somebody for the cattle issue. Please have advisory for receiving health certificate from all dairy products coming from USA or South America. There is a problem. So these kind of approaches will have to be basically put forward, planned, strategized. It's not one man show. It's a very comprehensive group, and efforts need to be done before we reach to the level where you can do sequencing within a few days and you take a decision.
Completely agree with what you said, sir. Lastly, can you shed some light on the importance of communication, especially the communication within the community in such events, even they occur in another country far away from Pakistan, but there is always a panic. So how do we can control there's some strategies that are helpful in communicating with general public, not only in such a outbreak, but in even bigger outbreaks or epidemics or pandemics?
This is one of the major issues that we faced in 2005 outbreak was in poultry, and people stopped eating poultry. So poultry business was gone down within a few days. So we had to take a lot of measures to activate and explain that eating cooked poultry does not transfer H5N1. But you can imagine the fear. Now with this cell phone in our hands, it could be help, it could be a danger as well. The information spreads like wildfire. I think it's very, very important to have very strong information setup to preemptively clarify things in people's mind. It's no way to create fear like in America, people stopped taking the milk of this, dairy milk and dairy products. First thing they did was they tested the presence of virus in the milk and pasteurization effect. So they were lucky that pasteurization, in fact, killed the virus. That's a separate story whether it transmits from drinking that contaminated water or milk or not. Same can go wrong over here. And our people are, they love too much and they hate too much as well. So they will overreact. So a good strategy would help us overcoming these kind of fears, I guess.
Thank you, sir. I think there is one question. So we have a hand raised from Muzammil Said.
It's amazing to see you again after a long time, Khalid sahab. Last time I was listening you in a poultry conference at UF, which was online. So nowadays it's also another informative presentation. Thanks for this. Secondly, I wanted to intervene. Why not we have such a compact system so far in 2024 as well? We didn't have any sectarian boundaries to detect these kind of diseases in our local farmers. Because of that, we are unable to tackle this problem so far.
Because if I understood you rightly, I think what your concern is that we need to have a system, I guess you said, system of communication. If you mean that there should be a system, there is a system, although it's weak, but there are, like the government of Punjab yesterday, the DG was telling us that they have 35 laboratories.
I wanted to mention that there is no input from the government so far. It is not interested in that area to protect our business, especially like with the poultry and the cattle. But there is a sustainable amount of this kind of viruses, especially influenza and other things that if it's not safer in the America, then here also we are also prone to that. So why not we have so far biosafety premises for this, even in this late age of sciences?
I think you have a point, but whatever happening right now is since the outbreak of H5N1, in those days, even we were more focused on disease control rather than developing, say, the systems that when the disease come and we are ready to accept it, ready to handle it. There's so many issues in countries like us. We just deal like there's a COVID, everybody is after COVID. There's like hepatitis, all the money transferred or moved. So we are poor country and we have limited resources and we have very, very short-lived priorities. So let's accept it. But I think whatever is available is that luckily we have experience of handling H5N1 of almost 10 years experience. And luckily the poultry sector is pretty, independent. They are handling things pretty nicely. I'm more worried about the dairy sector. The small farmers, 4 animals, 2 animals, so disease gets out of hand, we'll be in trouble. So let's communicate with the policymakers wherever I get chance, and I will try to share my thoughts. And we try to improve the system. We're living in it, and we cannot have an ideal system prepared for us. We have to contribute, make efforts, and one day I'm quite sure that things will improve.
Thank you so much, sir. Is there any other question? So if not, sir, we will conclude this webinar, and I would like to very sincerely thank Dr. Khalid Naeem Khwaja for taking time out. It was a 45-minute webinar, but sir, I think we have spent like 1 hour, so it shows the importance of this topic, and thank you so much for presenting. I think this was definitely quite valuable for all the participants. So thank you so much. And for all the participants, really grateful to you. And we look forward meeting you in another webinar. Thank you
very much. Appreciate it. Okay. Allah Hafiz.